Okay, hi everyone. My name is Nikolaj, and as mentioned, I'm a recent addition to CEH. So I started my postdoc in the beginning of March, and I'm going to be working for the next 2 years as part of the BarleyMicroBreed project in the group of Christopher Barnes. And the title of this, just very kind of brief presentation we're going to be having, is breeding for companionship in barley, optimizing the Holobiont functioning for enhanced drought tolerance. So, since the onset of Western agriculture in the Fertile Crescent 10,000 years ago, barley has become a widespread crop, one of the most widespread cereal crops. And during the domestication of the wild ancestor of barley, Holobiont spontaneum, there's been an ongoing selection of traits to tailor this crop for our ecosystem and for our production practices. During this selection, there's been a loss of traits, of course, genetic diversity and traits. Amongst these are robustness and abiotic tolerance that is inherent to the wild ancestor, and the consequences of that is something I'm going to be talking just very briefly about now. So in Denmark, we test the cultivars that are predominant on the market, and also new cultivars in registration every year at multiple locations as an adversary service for farmers. And what you see here is the spring barley yield of the spring barley cultivars tested from 1995 here up to last year, 2022. And each dot of this represents a mean yield for one of these cultivars. And during this period, there's been a considerate increase in yield. But most importantly here is are these 2 years, so 2008 and 2018. So these 2 years were characterized by very severe onset of drought. And as you can see, the entire panel here is showing a very considerate reduction in yield, more than 20% decrease in yield. So this emphasized the need for improving the drought tolerance in the elite cultivars of barley that are currently present at for farmers to use. And we therefore need to address this due to the events like these, or years like these, are going to be more frequent in the future as a result of climate change. So in order to ensure crop productivity and food security, we need some cultivars that can adapt and perform under these more dry conditions and drought conditions that occur. So in light of that, the BarleyMicroBreed was initiated in the end of last year. It's going to stretch for 5 years. And the objective of this is primarily to improve crop resource use efficiency, but also the interaction of barley roots with the indigenous soil microbiota to improve abiotic tolerances, to make them more climate resilient, you could say. And this is the larger scheme of the BarleyMicroBreed project. I'm not going to be diving into it. I don't really have the time for that. But it's a Horizon Europe project that involves 8 different institutions in 6 different European countries. And I'm going to be working on just a small subset of this scheme. So the plant genome of these barley, and very brief on barley genomics. So barley is a diploid grass species. It has 7 chromosomes, haploid genome size of 5.1 gigabase, and approximately 40,000 genes. The genome is extremely repetitive, with more than 80% of the genome composed of repetitive elements. It also has very low frequency of meiotic recombinations in large pericentomeric regions. So these kind of features of the genome has impeded the development of high quality reference assemblies until 2017, where the first one was published. And after that, several reference genomes have been published, and also a pangenome in 2021. So in light of that, the core of the work I'm going to be doing for the next 2 years is genome assembly of 50 barley cultivars. So that's going to be a selected subset of a larger collection that is being phenotyped and studied in the BarleyMicroBreed project. And we're going to be using, in order to bridge this large repetitive content in the genome, we're going to be using long read nanopore, Oxford nanopore technology with a combination of Illumina short reads to kind of correct some of the errors that might be remaining in the assemblies. So Oxford Nanopore Technologies relies on very different approach than other sequencing technologies. It relies on the use of a nanopore embedded in a membrane. And when the single strand of DNA passes through this, shift in electric current is measured in the membrane. And that, on basis of that, the bases are being determined using some processing of that data afterwards. So using algorithm to kind of decipher that shift in current. And there's been done a lot of improvements on that in recent years, especially in the new generation, the R10 generation of chemistry. So new type of core that has two sensing regions here, instead of the R9 generation just above. So this has kind of improved the raw read accuracy from maybe 97 to 99%, primarily due to the improved ability to call homopolymers. And in recent years, just last year, a publication was put in Nature, where they showed the new generation of nanopore for in bacteria, and they could conduct an assembly there without the use of the need for polishing using short reads. So hopefully we'll see something very similar, that we can go directly using the nanopore data without the need for successive polishing steps, and still get a very high quality reference assembly. So we'll see. But so the assembly is going to be conducted in two parts. The first part is going to be in a comparative study, where we're going to be evaluating long-read sequencing technologies in Barley. So here we've sequenced 3 accessions using both the approach we're going to use for the entire panel, so Oxford Nanopore Technology and Illumina, versus PacBio, to see how they perform in terms. And the study has been done like that actually in 2021, but then they used the R9 generation. So hopefully the new generation is much better. So we'll see how it performs. So in the end of that comparative study, we hopefully will be developing a pipeline. I'll be developing a pipeline for assembly using SnakeMake Workflow Manager, so that we can funnel the remaining data from the 47 cultivars into that, and then end up with 50 high-quality assemblies that we can then use for developing a pangenome. And that pangenome should then be used for identifying structural variations in this panel. So that's both single nucleotide polymorphism, SNPs, insertion deletions, copy number variants, also inversions, duplications, chromosomal rearrangements, and all these structural variants, that catalog will then be used successively to try and link that to difference in phenotype. So it will be used for association genetic studies for gene identification. And that will also be done multi-omics and all kind of different neat stuff at the end of this, but sadly that will not be on my plate. I think another postdoc will be recruited for that task. So in the end, this will lead to hopefully identifying a gene or genes that are very important for barley in order to improve its drought tolerance, and also interact with its with the indigenous soil microbiota, so that we can integrate that into new elite barley cultivars, and thereby improve the drought tolerance in the barley cultivars that are available for farmers, so that they can keep on producing and increasing yield despite these more frequent occurrence of drought events. Yeah, that's kind of the broader scale of it. Yeah. So if there are any questions.